First of all, good morning, everyone. Welcome to attend GenCefe's second webinar. We are a professional team of DNA, RNA gene synthesis and protein expression services. I'm the host of today's meeting kickoff. It's my great honor to see all of you here. Before we kick off, I'd like to take just a moment to introduce today's speaker, Dr. Jerry Sun, R&D Director of GenCefe. Dr. Sun received his PhD from Virginia Tech, USA, and has worked for many well-known companies in the industry. He is an expert in directed protein evolution, gene transformation, genomic, metabolic pathway synthesis, and R&D of deep proteins. In recent years, he has also made a breakthrough in gene synthesis automation. I hope you will enjoy his lecture. And if you have any questions during the presentation, please feel free to interrupt. Please keep your microphones muted when you are not speaking to minimize background noise. Thank you for your cooperation. Okay, now, Dr. Sun, are you ready? Can you share your screen and get started?
Okay, hello everyone. Thank you for coming to this my talk this afternoon. And in this talk, I'm going to talk about synthetic gene libraries and its applications in the study of proteins, antibodies, structure, and function. In this talk, I firstly will give a brief introduction of DNA library. Then I will talk about the DNA mutant library, give introductions, then its applications and quality specifications. Also, the mutant library categories and our service specifications. And also, I will talk about a bit about sgRNA libraries. And last, I will give an introduction of GenCefe tech. So firstly, I will talk about synthetic gene library. So first, I will give a brief idea of DNA library. So DNA library is of DNA. And instead of a single sequence that gene we usually use, so DNA library is a mixture. It contains a lot of different DNA sequences in the sample. So it's not a single sequence. This sequence usually comes from have common characteristics. Sometimes it comes from single organisms or derived from a single parental sequence. We can briefly group the DNA library into these categories: DNA level research, RNA level research, and protein level library. So the DNA level libraries, we can investigate the DNA's functions, investigate the promoters, terminators, and any cyst function elements. And also sgRNA library. sgRNA library is used to investigate the gene lookout and lookin in genome-wide scale. And RNA level library contains cDNA libraries, which is derived from mRNAs, and the protein level library, which is the major part of this talk. I will talk about the mutant libraries, either random mutant or controlled mutant libraries. When we talk about library, we have some key factors or key indicators that are to evaluate the quality of a library, and include the capacity, library capacity, accuracy, coverage, and bias. I will talk more about these indicators later in my talk. So the synthetic DNA library, in these years, the development of synthetic gene techniques will make this more and more easy to create synthetic libraries. Previously, we made libraries from, for example, from organisms, but we have a lot of experience in production the synthetic DNA libraries. So here we talk about synthetic DNA library. It mainly refers to such direct mutagenesis libraries, random mutant libraries, degenerate libraries, and also sgRNAs. For mutant library, I will a brief idea of how amino acid how diverse a protein can be. So say you have a protein that composed of 100 amino acids, so that sequence diversity would be 20 to the power of 100. That equals 1.2 times 10 power 130. That's a huge number. If you assuming that there's a mixture contains all of these properties, possibilities, and one possibility has one factor, the mass of this mixture would be greater than the mass of the universe. So this is an incredible large number. So it is impossible to design a gene library that includes all of the possibilities. So in the practice, we need to try to minimize the amino acids that we investigate. So here is an example of the use of the mutant library to modify the enzyme. So look, here is the luciferase. Luciferase was widely used in the kits to track the protein movement in cell. So previously, this enzyme was the Y type was very weak. It gives very weak signals in the experiment. There's a company that tried to make mutant libraries and did a large-scale screening of this protein, and finally they got a mutant luciferase, which has 100 times higher activity, and with reduced molecular weight. So this is a very successful application of the mutant library used in the modification of protein activity. This is another example of the mutant library used to modify enzyme activity. So there's a company that used the library to try to increase the activity of an enzyme. This enzyme is a glucoamylase, which is used widely in the production of in the process of starch degradation. I found that they got a 10% increase of activity, which is a very significant increase in the industrial production. So I looked for the examples and found a lot of articles making mutant libraries. Here is an example. Attack polymerase is widely investigated. So I list here some articles. So in these articles, random different kinds of DNA library was used. They include random mutagenesis libraries, site-saturated mutant libraries, and also rational designed mutant libraries. And the phenotype they are looking for include the activities and resistance to inhibitors and other beneficial phenotype. Listed here is some examples of industrial used enzymes, which is widely used. The amylase and the glucoamylase were used widely in starch degradation, and the cellulose and the lipids were used in the food production. So here, the random mutations libraries and the saturated mutagenesis libraries was used. And the phenotype they are looking for include the thermal stability and the activity increase. Another important application of mutant library is to modify the antibody. It uses the one to so here is some examples of the antibody used. Because the antibody is widely investigated, the structure of is well known. So the modification usually focus on the so here is the substitution mutant and libraries and saturated libraries, and also rational designed libraries. They are looking for higher affinities. So there are a lot of successful examples of libraries used in antibody. Next, I'm going to introduce some concept of key indicators of library, which is used to evaluate the quality of library. So here is the concept of theoretical library capacity and the actual library capacity. The theoretical capacity means that there's how many different numbers of variations in the library. It is the maximum library capacity. So here, if you have a sequence listed here, you have 4 ends in the sequence, so that diversity would be 4 to power of 4, so equals to 256. So there are 256 possibility sequence. This is a theoretical library capacity. And there's also actual library capacity, which is how many clones I can got from the plate. This capacity was calculated from the how many clones we can got. So there's a contradiction between the theoretical and the actual capacity. So in the design of the library, we're trying to include more different molecules, but in the practice, because of the limitation of the cloning techniques, we cannot do a library with too many variations. So, with this, at this time, we use E. coli cloning technique and normal biological techniques. We can do actual library that is less than 10 to power of 8. A typical library usually contains a capacity of 10 to 4 to 10 to power 7. That's a typical actual capacity we can do. The next idea is accuracy. Accuracy means that how many sequences in the library is correct. That means how many what the percentage of the sequences are correct. Some of the sequences may be wrong or nonsense. So in this case, we have a library. We sequenced the 24 clones, and 22 of them are correct. So the accuracy rate will be 91%. The next indicator is the coverage, which means that the coverage can be simply understood as the library completeness. So in this library, does it contain all of the sequences that we designed, or if it missed some of them? So here is an example. In this library, there is 1522 sgRNAs, and the NGS sequencing result gives 1519 sgRNAs. So the coverage would be 99.8%. A typical library will have coverage. It depends on the size. If a library with a small size, less than 1,000, we usually give 100% of coverage. If a library, a big library, is having a capacity more than million, so we will give a small coverage number. So this depends on the size. Then the bias, that's another indicator of library. Bias means that bias means that all of the sequences have approximately equal numbers of molecules in this library. So is there any sequence that takes too small, very little sequence in the library? So this will affect your next step screening. If a sequence have two more molecules in the library, you may miss the sequence. We have a calculation equation. I will not go to details of this. Then I talk about randomized mutant libraries. So here is an example of randomized mutant library. In each sequence, there is one or two mutated nucleosides. This library is usually used when you have very little knowledge of the protein. This is accomplished by usually by error-prone PCRs or by DNA polymerase with very high mutation rate. So you so it is used to study the protein structure and function, and also antibody humanization. Randomized mutant library can be divided into single point library or multi-point library. Single point library includes the RNA scan library or annual glycine scan library, and a single point random mutation library, which is usually did by PCR. And the multi-point random mutation libraries, in which there's more than one mutation point in each molecule. Here is the scan library. In each sequence, there is only one amino acid was substituted by alanine. And this library we can deliver either single clones or a mixture of the all of the sequence as a mixture. That depends on your choice. And next is the degenerated mutant library. This is widely used to investigate protein function. So in this library, some of the amino acids were degenerated to other 20 amino acids. So this is a real example of degenerate mutant libraries. In this case, you can see many amino acids was substituted by AMB, which code for 20 amino acids, any of the 20 amino acids. So in this case, the theoretical capacity will be, in this calculation, it will be very large. This number is very large. So you have many amino acids substituted. So in the design of mutant libraries, there's many, we need to, usually the region of degenerated region should not be more than 100 base pairs. And between the two regions, there's at least 20 base pairs between them. That is for cloning purpose. And there's also other requirements. Here's another real example of the degenerated library. In this case, there is this. All of this region are degenerated. So this is a very huge library. And the next controlled library. Instead of degenerated amino acids to any one of the 20 amino acids, you can degenerate the amino acids to some of them. You can choose any combinations of amino acids that you can degenerate. So here you can see, you can substitute the D amino acids into this amino acids, and also the R. So this will make the library size much smaller to facilitate the next step of screening. So here, this is a real example of the controlled library. This is from a patent. So here you can see it degenerated the amino acid to some of the amino acids, not all of the 20. In this case, the capacity or the variability is 7.8 times 10 to power 5. So it's close to 1 million. So we provide random different types of library construction, include randomized mutant libraries, site-directed mutagenesis libraries, and degenerate mutant libraries. In order to make the library, you need to have the Y-type gene. If you have the Y-type gene, you can ship to us. If not, we can synthesize the Y-type gene. And then based on this Y-type gene, we can do the mutations. We can deliver the library, deliver DNA, either the DNA or the cloned library in bacteria. Yeah. If it's a cloned library in bacteria, in plasmid, the typical library capacity will be 10 to power 4 to 10 to power 7. If it's a PCR product, the library capacity can go up to 10 to power 11. Next, I'm going to talk about sgRNA libraries. sgRNA library is another kind of synthesized DNA library. Because in these years, the development of DNA synthesis techniques, especially the GenCefe synthesized technologies, it makes it possible to synthesize a lot of oligos on a chip. The number can be tens of thousands different oligos in a single chip. So sgRNA library, we synthesize a lot of oligos on a chip simultaneously, and then clone the different clone the library into the vectors, then pack the virus and infect the cell, then do the subsequent screening. This is used to do large scale gene lockout and lock-in. For sgRNA libraries, we can design the sgRNAs and also construct the sgRNA library and make the plasmid and do the quality control. sgRNA library quality control usually need to add NGS to sequence. If you want the NGS sequence to as a quality control, we can do it. Next, I will talk about give a brief introduction of GenCefe biotech. GenCefe biotech was established in 2022 in China, Wuxi, China, and started its branch in California in 2021. We focus mainly on DNA, RNA, and gene synthesis. So here is the headquarters of GenCefe in Wuxi, China. So we founded in 2022 and established the oligo synthesis and the single sequence platform in 2022. And we established the RNA synthesis platform in 2021. And we built our AR tool set in 2022 and also cell-free protein express platform. This is the main service we provide in GenCefe, the oligo synthesis and modification. A gene and plasmid platform that provides genes and plasmid, large-scale plasmid preparation, and also cell-free protein expression platform, and also the AI-aided designs of sgRNAs, sRNAs, and libraries. This is some snapshots of our lab. These are some of the intellectual properties that we have. And this is our headquarters and branches in US. We also have offices in the main city of China. These are our partners, academical and industrial. These are some our citations, published citations. Yeah. If you have any questions, you can go ahead to ask.
Hi, this is Azhar from India. Yeah.
Hello? Yes.
Hello.
Mm.
So I have one question. So you provide this synthetic library in as a gene fragments, or you will provide in a vector?
Both are okay. We can provide the gene fragment, and also can provide them in vector, grounded in vector. Yeah, both of them are okay.
Okay. So for antibody synthetic library, so you provide the naive library and or it's a randomized library. So do you do the mutation in the CDR region or entire antibody variable region you do the mutation?
I mean, if we can do the mutation in a variable region, I didn't get your question. I'm sorry.
Azhar, can you repeat the question, please?
My question is, do you provide it's a naive library, or you provide any randomized library?
Yeah, randomized proper library can be used in antibody modifications and immunizations. Yes. And
okay. So my question is, so do you provide because do you need a wild type gene? For example, if you have to screen an antibody library, so do you do the mutation in the wild type gene, or you provide this the big library you can screen for primary screening?
Usually you need to have a wild type antibody. We based on this antibody then design the mutant libraries. Because usually the wild type antibody have some affinity to the antigen. You need to, if you want to increase the affinity, you can do the modifications. But recently there's also techniques that design the antibody in silico. In this case, you don't need the wild type antibody. You can just design from zero, I think. Then do a large scale screening. That's also a process.
So do you provide the naive library also? So you provide the naive library also. Is that right? If I'm not wrong.
You said name the library. What's that?
Okay.
It's a naive library. Means without not having any gene. You don't know the prior information of any gene. So just take any random sequence and just do the random mutations and just screen. Like synthetic library. In synthetic chemistry, they do the random library. Like that, you do any library?
You mean you don't have the antigen?
No, actually, for example, if you don't know anything about, for example, you don't have any wild type gene, still you would like to screen. So you can make a random mutations, and then it's a in huge number, and then you can screen. So that kind of library do you provide?
You mean a fully synthetic protein library, correct?
Full,
Yeah, exactly.
full synthetic. So you don't have a wild type antigen antibody. So you want to design an antibody. Is that correct?
Yes, exactly.
Yes, we have that's a good point. Yes, recently there's a lot of studies on this. Some companies start to design the antibody in silico, in computer, then make based on antigen, make computer model simulations, then design the antibody, then do a lot of then express a lot of this design the molecules, then do a large screening. This is actually a new method to obtain a good antibody. It's fully designed. There's no wild type parent or antibody available. Is that correct?
Yeah, fine. Thank you.
I would have also a question regarding the DNA synthesis. Are you able to do triplet-based synthesis procedures where you, rather than mutating single nucleotides, where you take as building block triples, codons already? Okay. The DNA synthesis you usually do with oligos and with single nucleotides to be added on. And I'm just wondering, or would be curious, if you have strategies developed where you take three
I get
people-based synthesis.
I got it. Yes. The answer is yes. We can do that. That is trimers. I think is.
Yeah, correct.
Yeah, we can do synthesize that based on trimers. Yeah, we use trimers to build our libraries. Yes.
That's okay. Thank you.
Thank you, Aja and Oliver, for sharing your perspectives. Is there other questions? I think it's a good opportunity to exchange your opinions and discussions.
Oh, I liked the introduction you gave and the overview about the technologies. And I mean, it always depends on the project at the client is always a black box. So, but it was very informative what you, to get that intro. So thank you very much for that.
Thank you everyone to join the meetings.
Yeah, if there is no other questions, as we approach the end of the meeting, it's time to say goodbye. If you think of anything after the meeting, please feel free to reach out. Thank you all for coming, and we look forward to seeing you in our future webinars.